So I got one more video for this. But I'm going to kind of remove. I'm moving away from phylogenetics to a little bit of a whole genome analysis to some degree. I guess this demonstrates a couple of things. Whenever we do have a whole genome to work with, we can also work with coordinate information, right? Whenever we do look at a giant genome file, sometimes we're looking at the DNA sequences, and sometimes we're more interested in the coordinates, because the coordinates can tell us a lot of information about gene duplications and a handful of other things, where a gene is, what its neighbors are, is that gene order conserved, do we see it, do genomes look mixed up? Things like that can be answered from coordinate information. But in this tutorial of playing with coordinate files, we're also going to learn another special character and a way to use grep. I think overall we'll probably learn about three new commands here, to not only just demonstrate the power of Linux, but also demonstrate analysis of coordinate files, or at least some basic analysis of coordinate files. So whenever I'm working with any good genome file, or looking for any good genome file, I'm starting on Ensemble, right? That's just kind of a go-to by this point. I think it's just nicely curated, and most of the time DNA data is pretty easy to access on Ensemble. We'll browse the NCBI shortly, but Ensemble's where it's at, especially for vertebrate genomes. If it's here, this is where I go. So I'm going to go get some coordinate information, and I'm going to get. So I'm going to go to. I went to downloads. Now I'm going to go to FTP download. We've been here before to get CDSs, right, to demonstrate what we can get off of Ensemble. But another thing we can get off of Ensemble is the gene coordinates, or the gene set. So we see through human, working with human is nice. It's probably what the better curated genomes, most popularly used. But yeah, so we have a whole genome column, a cDNA column, CDSs, blah, 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 and then we get a gene set. Now this GTF and a GFF, these are file formats that that describe coordinate information. I tend to favor the GTFs over the GFFs, really for only one good reason. It's because I really only work with GTFs. That's it. Not for any other special reason. There's reasons to use one or the other, but traditionally I use the GTF. So I click on the GTF. I'm going to get this. Hopefully you're reminding yourself how we get big files from the internet. Just right click on the file we care about. In this case, it's going to be the this Homo sapiens 103 GTF. We don't want the ab initio. We don't want that one. We don't want the chromosomes. We want the raw GTF, because if we just see the CHR, this is the coordinates of just chromosomes. However, not DNA genomes don't get assembled to the level of a complete assembly, chromosome 1 start to stop, chromosome 2 start to stop, chromosome 3 start to stop, etc. What we get are scaffolds. And so usually the less processed genomes are more fragmented, and they may have hundreds or thousands of scaffolds that represent a contiguous sequence, but we don't necessarily know where it came from in the genome. We don't know if it came from chromosome 1 or if it came from chromosome 20. We just know its scaffold number in. So if we're working with a lesser processed genome and we get the chromosomes, there may be nothing or very little in that file, and we may be missing half of our data because half assembled the chromosomes and the other half it's someplace. We don't know where it goes. So this GTF.gz is going to have everything on completely assembled chromosomes and also the unplaced scaffolds. So I'm going to get that address. I'm going to wget that link. I'm not going to do this because it takes a little bit long and I'll just be sitting here for 5 minutes. I've already downloaded this, but nonetheless, I'll hit enter and wait until that's downloaded. But I already have this done. I'm just going to move it into a location where I can play around with. And I want to keep playing with my test, playing around the test directory because I don't want to muddy up all my other directories. So I'm just going to make a quick directory for Philo stuff. And I'm going to move everything into Philo. There we go. And I'm going to make another directory for GTF and move, where do I have this? I have that here on my home. So I'm going to move this home, this Homo sapiens, into my test GTF. So just have a clean directory. This is the only file I'm looking at, and all is well. So this is, we're reminding ourselves about Gzip files. We've seen them before, but we haven't seen one that looks like this. So first off, just to remind ourselves about Gzip files is I can't inherently look at it. So if I try to cat it, if I try to head this file, I'm just going to get some binary garbage, right? That's related to putty stuff. I just can't see it. It's garbage. If I wanted to look at this file, I would have to do a gunzip. However, I don't necessarily have to use a gunzip to look at this file. I can use another command that's zcat, and zcat is equivalent to cat except for Gzip files. This is a Gzip file, so I can zcat this. So I'll just quickly let this go for a second. I'm going to Control Z when this is done. So I'm going to zcat it, and we see a lot of garbage, but nonetheless, it's text garbage. It's not binary garbage, it's text garbage. So this is, we can read it with a zcat. All right, so again, what's in a GTF file is largely gene coordinates and also additional descriptors about that gene. So additional information about that gene. Before we look at this further, I'm going to introduce a new special character. This special character is a vertical line. In Linux, it's called pipe. I think in general, it's called pipe. And so this character is below the backspace or the delete button. And there will be a / and this pipe on that button. So it's one of the most furthest right buttons. And you shift that button to get the pipe. And so how this pipe works is it sends output of one command as input to another command. I mean, that's effectively what's happening. But we have to, whenever we're working with zcat, since we just can't cat the file or less or anything like that, we kind of have to use this pipe a little bit more creatively. And so, for example, if I'm going to do this literally, I'll do a zcat on the file, and then say I just want to look at the first few lines of it, right? That's head. And so I can pipe that output to head. And so the output of zcat is going to be the input of head. I hit that, and now I only have the first 10 lines of this Homo sapiens GTF file. And there's a lot of mess in a GTF file. So much mess that it definitely deserves to be made fun of. But let's try to look at this in Excel, because it's a little bit more polite to look at this in Excel than anywhere else. And I'm just going to get, say, the first 20 lines. Right, there's just a lot of wrap text in here, and it's definitely certainly a pain to look at. So I'm going to have it as tab delimited in, or space delimited in Excel. And so now this is a lot easier to read. So what's happening in a GTF file is that the first column is going to be the chromosome number. The second is going to be a gene prediction method. Further, now this gets a little bit annoying, but here we go. We have a whole lot of columns over here, and these columns are they're semicolon delimited. I imported this into Excel as space delimited, so anytime I have a space, it's going to turn into a column. But nonetheless, all of this would be considered one giant column that's delimited by this semicolon. And so in this, I can have a gene ID, a transcript ID, and exon number and additional properties about this gene. So GTFs are a little bit awkward to read, certainly with the naked eye. Computers can read this a lot easier, but with the naked eye, we'll see a few things in this. So the first line of any new gene is, I guess we can call this, I don't know, biotype, I don't know. But this gene starts at base pair position 11,000 on chromosome 1 and ends at base pair 14,000. Very short region, so 3,000 bases, and it's mostly contiguous. And this plus means it's in the forward orientation given the chromosome number or the chromosome orientation, and a minus would reflect it's on the opposite strand. Now here's where it gets a little weird, because we define the full gene start to stop, but we have to kind of remind ourselves that every gene can have multiple transcripts. And so in this GTF, we have this gene coordinate, and then we also have a transcript coordinate, and where all of those exons for that specific transcript occur at. And so the start and the stop will be the same. So we have a start of 11,000 and a stop of 14,000. So our transcript start and stop is the same, but our exons, our first exon starts at 11,000 or 11.8 thousand and ends at 14.4 thousand. And so all of these rows describe, really all of these rows here describe the exon structure of this first transcript. And again, we have multiple transcripts. And so we have another line of text that describes the exon structure of another alternatively spliced or another possible transcript. And we know that all of this stuff is referring to the same gene whenever we look at the gene ID. So our gene ID is the same throughout this entire region. So all of this information is describing one gene, particularly the gene coordinates, the transcript coordinates, and the exon coordinates. And then when we're done with that, our next gene will be in place. And so it's kind of, well, this is ugly. This is ugly, and this is ugly. I know, I can see that it's ugly. You don't know that's ugly, but I can see that it's ugly. And partially because it's ugly is because it's right next to the telomere, right? We're only 10,000 bases into chromosome number 1. We're probably even, we're right next to a telomere. So the annotations aren't going to be all that great. Nonetheless, we do see multiple genes. So here's our first gene on this chromosome. It's DDX11L1, first gene. And our second gene is WASH7P. That's our second gene, right? So all of this information is regarding this first gene of our gene name, right? All this gene name information, gene ID is the same. Now our transcript information changes, right? Here's our transcript ID. And so there's the ID for this first transcript, and there's the ID for the second, alternatively spliced transcript, right? All this stuff's here. And this just gets even more complex as we scroll through this GTF file. We see all types of stuff here, right? We see a gene name repeated. We have exon numbers repeated as we should, 1, 2, 3, 4, 5, 6. And we get this biotype. And so this biotype kind of will tell us what this region is, right? So here in the second column, it's just calling it a gene. But whenever we actually look at the biotype, it's saying transcribed, unprocessed pseudogene. And it's not necessarily surprising to see a pseudogene near the end or around telomeres. It's near telomeres, and hopefully we're all familiar with what a telomere is. It's just it's a repeat on every single chromosome. Linear chromosomes will have this region where it's like 5 or 6 base pairs just repeated over and over and over and over and over again, like thousands of times to maintain chromosome integrity, right? Every time chromosomes divide, we lose a little bit of that telomere. So stuff near telomeres, probably going to be garbage. And in this case, certainly the first two genes, or the first two genes that neighbor the telomere, are transcribed, unprocessed pseudogenes, which means that it is transcribed, but it's unfunctional because there's probably either a frame shift mutation that's throwing off the whole reading frame, or there's a premature stop codon. So it can only be transcribed to a point, and the product is going to be garbage. Yep, and then a whole bunch of other stuff. But either way, and quite honestly, the only reason I know this is because I've looked at several GTF files. And so this will just repeat all the time, right? From chromosome number 1, that's going to be the start, that's going to be the end, and orientation. So those are pretty important columns: chromosome, start, stop, and orientation. That's important generic information. The gene ID is kind of important, and the biotype is pretty important. And so in this GTF file, we can see pretty much anything that exists that's not an intron, that's not a TE, be annotated. So protein coding genes are annotated. Pseudogenes are annotated. tRNA loci are annotated. Our ribosomal RNA is annotated, right? All of this stuff, even the non-protein coding stuff, will be annotated in a GTF. It's just finding that will probably take a little bit of time to do. Or we can play around with grep that we've seen before. Yeah, so this sucks. Like, looking at this in Excel is way easier than looking at this in the terminal, no doubt. But we've introduced the pipe. And so let's play around with trying to pull information out of this GTF file with grep. So let's just start with RNA. And I don't want this to run all day. So I'm only going to look for the first 10 instances that we see RNA in here. And let's see what we see in here, this kind of garbage. Okay. So here we go. So this all is all linked to gene number, or the first thing that we found, the first line that we found. And what we found here is a microRNA, right? So here's an annotation for a microRNA. And that seems to be common. So chromosome number 1, there's another microRNA. It's just a derivative, still the same RNA, just a derivative. Bunch of microRNAs. Oh, now we have a link RNA. So that's probably a new thing we've never heard of, but this link RNA, all of this stuff here, a link RNA that stands for long non-coding RNA. And this is about 2,000 bases of a single region that's going to be transcribed, but not translated and serve a purpose other than an mRNA or a tRNA or a ribosomal RNA. It's undefined. We know it's transcribed. We know it's going to be an RNA, but its function is. But yeah, that's about 2,000 bases. Let's see if we can find a tRNA. Let's see if that I can grab that out. Nope. It doesn't look like I can because nothing's popping up. No lines. Oh, we do. Oh, we do. Look at that. Let me do that again so I can see what I'm. All I see is so far is just the empty tRNA, and that seems to be true. And so that tells us that it's on the mitochondria. So MT is going to be on the mitochondria. There, its start and its stop, and its biotype is an empty tRNA, right? So that's annotated in the mitochondria. Furthermore, we can see everything that's on the mitochondria if we grep out MT, right, for anything mitochondria. And I'll take this one step further. Instead of heading it, I'll pipe all of this information to less, right, so that I can read through it and kind of control what I see. Oh, something stupid happened. Grepped NT less. Oh, I know why that happened. Because MT pops up all over the place, right? MT, right? So I grepped out every line that starts with MT or has MT in it, not necessarily mitochondrial line. So that was a failure. And so let's say I want to just look for the protein coding genes. As I mentioned, the easiest stuff to understand in a genome is the protein coding. And I know this because I've looked around enough at this that a protein coding gene with an intact one that's functional will have a biotype that is protein coding. And I'll head that. Okay, so yeah. Chromosome number 1, gene ID, we call it OR4, it's just an open reading frame, or OR45. That's probably an olfactory receptor. Could be, probably, I don't know. But yeah, protein coding. There's our biotype protein coding. But nonetheless, so let's just kind of look at this for a second in, let's see what this looks like in Excel. All right, so now we get some more information because this is a protein coding gene. The CDS, like the coding sequence itself, right? So our transcript information is defined, where the transcript start and stop is, but so is the CDS. So the actual coding sequence. And so the first, from what we can tell, the first protein coding gene on chromosome number 1 exists at base pair for 65,000. And it's in the forward orientation. It's going to have several variable exons, transcripts, etc. But yeah, And so since this is a protein coding gene, we also annotate the start codon, like where's the actual start codon. And I bet if we take this even further, we should likely see the stop codon. So let's see if I can highlight all of this and see if we can get the complete information for this protein coding gene. Oh. Yep, here's our stop codon. But even further, right, we can get annotations for the 5' UTR. It probably means we can get the information for the 3' UTR, etc. So everything pretty much gets annotated for a protein coding gene. Start, stop, exon starts, all the exon IDs, etc. And so the thing is, what makes this a little bit annoying is that we do have multiple pieces of information referring to the same region on multiple lines. That can be irritating to look through because if we just try to say grep out a gene ID, this gene ID is going to pop up for every single gene, transcript, CDS, start codon, stop codon, exon, 3' UTR, and 5' UTR. Way too much information. So it's not uncommon for me to just simplify a file, a GTF file, and just pull out all of the lines that are protein coding and have the word gene in it, right? Because that's telling me the exact, I know where it starts, where it stops. I don't necessarily care about the exon intron boundaries. I don't care where the stop codon is. I just want to know where this gene can be found in a genome. So if I want to say get that information out, I kind of have to be creative with my pipes. And I have to be creative with grep. So here's a nice command, a tricky command with grep. That's grep-v, a new option. This is grep-v is the opposite of grep. So if I say grep CDS, it's going to pull out all lines that have CDS in it. But if I grep-v CDS, then it's going to return all lines that don't have CDS in it. So with this information, I can say grep out specifically protein coding genes. But I don't want all of those lines that have transcript, exon, CDS, 5' UTR, etc. in them. So I can, for example, grep-v exon. But I can't grep out multiple. I can't grep multiple words with grep. I can only do one grep, one word. I can't do grep protein and grep-v exon in the same line. It has to be an individual grep command, all connected together with this pipe, with that pipe. Okay. And so I'm getting a little closer, but there's still all of this extra stuff on here. So there's the 3' UTR that I don't want. There's the 5' UTR that I don't want. Transcript stuff that I don't want. I did this once earlier, but the more I looked at this, I probably did something wrong. So I'm just going to exit anything that has transcript in it. This is probably a mistake, but it's okay. It's okay. Yeah, it's okay. Because now I've done that. Well, I wonder if I'm being redundant with the grep-v exon. Let's see. I think so. Yeah. So with this, whenever I grep for protein coding, but I grep-v transcript, I'm basically only going out to print the lines that define the gene region, right? Because I'm ignoring lines that are like transcript ID. And so I can save this to a new file that can be text. So for example, human let's just say gene.dtf, because it's still a GTF file. It's just I filtered this stuff out for only the stuff that I care about. And so that'll take a little while because this is a massive genome. It takes forever. And there we go. 3 megabytes of human gene information. So let's less this for a second. Let's see if I can read this. And it's a pain, but it's okay. But notice the information is still here. So all of this is one line. And so I should, if this worked correctly, then my gene ID should be different on every single line. And that seems to be true. That seems to be true. And my gene name changes every line too. So basically, I have collected and pulled out all of the protein coding genes and the coordinates thereof for this GTF, just by being creative with my grep and a pipe. And of course, I'm finishing off my command with an open caret to save that to a new file. Oop. Okay. And I guess the last thing I can do, the last command, kind of a new command for today, is an easy one. It's WC. It stands for word count. And so WC is just going to count the number of letters, count the number of words, and count the number of lines in a file. So if I do a double WC on human, it's going to tell me that there are 19,000 lines. I think 353, I don't know what this number is, and I think that's raw characters. That might be letter count, and this might be all characters in general with spaces. Oh boy, whoops. Let me confirm that before I tell you wrong. Number of lines, words, and characters. Yep, so this is lines, that's the number of words, and that's the number of characters. But right, so if every single line is a protein coding gene, and it's all the protein coding genes in the human genome, that tells me that there are 19,000 protein coding genes, unique protein coding genes in the human genome. And kind of interesting is this number keeps getting smaller. Like the better our annotations get, the better we are at identifying genes, the smaller this number gets. Right? We started, like if we went back to like Ensemble 1, we're currently on Ensemble version 103. But if we went back to Ensemble 1, the human genome, that draft is probably going to have 25,000 protein coding genes. But now we're down to 19,000, 19.9 thousand. It's just because this gets better. And it also tells us that it's tricky to identify what a gene is. Because they're broken up into exon and intron boundaries, just because a region may look genic doesn't necessarily mean it actually is protein coding. And so that phenomenon or that trend has been continuous among all vertebrate genomes, is that as we get more refined, what we previously called the gene turns out it's really actually not a gene. It never produces an RNA in any tissue that we've ever seen. So it's definitely not a protein coding gene. but no less. Yeah, so what we've done today is, I guess, our 3 commands is zcat that we've learned. We learned the pipe special character. We learned grep-v and word count. And so that's probably enough. I think that's enough. But whenever we have these massive files and we don't know how to code, just knowing a couple of unique tricks can help us filter files out pretty easily. So yeah, I'll leave it at that. Maybe the last thing that we'll play with. Let's see if I can. I don't think I will, but let's see if I can grep out MT now. So all I get, yeah, I still get this. Yeah, we have MT in some of these gene names, fine. But the last bit of our file does have the protein coding genes of the mitochondria. So there's ND1, CO1, CO2, ND3, ND4, and site B. So I think in total, I bet if I grep on this on a dash, yeah, it just gives me, oh God, almost so close. Just one stupid gene name. So close. Anyways, I can count that. So I'll just do a quick WC. And if I take into account one thing is mismanaged, I know I have 13 protein coding genes in the mitochondria, right? Again, not knowing really any coding information, just taking advantage of grep and pipe, we can learn a lot about genomes and what's where. All right, so that'll be it. Now for sure I'll call it. I'll see you all later.